Today we're going to cover Chapter 4, the functional anatomy of prokaryotic cells. The chapter does also go over eukaryotic cells, but our focus is going to be on the prokaryotic organisms. When we look at a prokaryote and a eukaryote side by side, oftentimes people see them as the same size because we're trying to emphasize the features of both. However, if we compare prokaryotes and eukaryotes, we see they're actually very different in size. This is a more realistic representation. So we see the bacteria, the prokaryote, is about the size of the chloroplast and the mitochondria. And that's going to be discussed more later in the slideshow. So when we say the term prokaryote and eukaryote, what does that actually mean? Well, karyon refers to the nucleus, and so when we talk about a prokaryote, that means that we're talking about something before the nucleus. The EU designation means true, and so when we talk about eukaryotes, this comes from the Greek word for true nucleus, and that's a basic defining feature of differences between prokaryotes and eukaryotes. However, when we look at what the comparison looks like, it's a little bit beyond just the nucleus or not. First of all, prokaryotes only have one circular chromosome. It's not in a nucleus, so therefore not in a membrane, whereas eukaryotes have paired chromosomes that are in the nuclear membrane, the nucleus. Prokaryotes in general have a smaller genome, so they do not have histone proteins that are utilized in eukaryotes to wrap the DNA around and cause it to be more condensed for fitting into a eukaryotic cell, and specifically into the nucleus. So prokaryotes, they don't need to have those histones, so their DNA kind of looks like a spaghetti mess. But it is just one big circular chromosome. Also, prokaryotes do not have organelles, and we see that a defining feature of bacteria is that they have peptidoglycans in their cell walls. This is going to be a major feature when we talk about our gram staining, and we'll see that in a few slides. Prokaryotes also encompass prokaryotic organisms in the grouping of the archaea. They have pseudomurine in their cell walls, and even though they look a lot like other bacteria, they're a separate category, as we'll see later on. Another major feature is prokaryotes divide very rapidly. Binary fission just means they split, so 1 becomes 2, 2 becomes 4, 4 becomes 8. We'll talk more about the microbial growth in the chapters next week, but suffice it to say, the division is different than with the eukaryote, going through the fact that they have things like a mitotic spindle to be able to separate their chromosomes that are going to be important in the division of the cell. So in summary, we see prokaryotes and eukaryotes, they both have DNA, but prokaryotes have this in a single circular chromosome. Eukaryotes have this in paired chromosomes. They have ribosomes for protein synthesis. They have cytoplasm, and they have to have a cell or plasma membrane. But prokaryotes are considered much simpler. They're smaller, they lack a nucleus, no organelles, and they are the most primitive of the cellular types. When we talk about prokaryotic cells, although there are a variety of shapes, they can mostly be synthesized down to 3 basic shapes. Things that are rod-shaped or bacillus, spherical or cocci, and we also have a variety of spirals that can be broken down into spirillum, which are kind of more of a looser curve, vibrios, which are just a single curve, comma-shaped, and corkscrew-shaped spirochetes. When we look at these in actual size, these are too small to be seen by the naked eye, and normally we have to use a microscope, and beyond the fact that we have to use a microscope, we have to specifically use oil immersion to allow us to magnify them 1,000-fold to be able to see. Their average size is about 0.2 to 0.1 or 0.2 to 1 micron, and times that by about 2 to 8 in their length. So here we see the basic sizes and shapes. The cocci can be in clusters or in chains. The rods kind of look like little Mike and Ike candies. The green ones in the lower panel, those are coccobacilli. They can't quite make up their mind whether to be a rod or a cocci, so they look a little bit more of a oval shape. On the right-hand side, we start to see our various spirals. The top panel, those are vibrios that are just a curved comma shape. The spirillum in the middle just kind of has a little bit of a twist to it. And then the bottom panel, the spirochetes, are really a corkscrew shape. There are some other unusual shapes found in nature. So these are usually more likely to be out in the environment, in ponds and various aquatic environments. So we can see star shapes like the stella and the haloarcula, but those are usually not clinically relevant. What is relevant is most bacteria are monomorphic. That means they have a single shape. That's important because they're going to look that way whether it's the first day or day 7 in their growth or during infection. So that single shape is important in our identification of the organisms. There are a few organisms that are pleomorphic that have irregular morphology, and oftentimes those morphology changes are signaled by changes in their environment. But by and large, when we talk about the shapes, a cocci is going to stay a cocci, a rod, and that is going to be important in helping us to identify the organism. Besides the basic shape, they can also have various arrangements. For cocci, they can divide along multiple planes. So if they divide along a single plane, you see things like diplococci, just 2 cocci together. Others may end up causing a chain. We refer to those as streptococci, strepto meaning chain. Then if they have perpendicular planar divisions, we see we can get things such as tetrads and a cube shape, sarcinae, and we also have grape-like clusters. Grape-like clusters are staphylococci, and we see that these arrangements often give rise to the genus names that we'll begin to explore throughout the term. For the rods, they can only divide along one plane. So we see most of the time the bacilli are going to be just single rods. They may form a diplococci, and occasionally you may see some streptobacilli, but for the most part, they're not going to have the diversity of arrangements that we see with the cocci. In the bottom panel, again, is that coccobacillus. Again, it just is a elongated oval, so it's between a rod and a cocci, but when push comes to shove, coccobacilli are referred to as rods, so don't be confused by that designation if you're looking for their gram reaction. So in summary, the basic shapes, we have our cocci, which are spherical, much more diversity in their arrangement. The bacillus, which is a rod, they can have diplobacilli and streptobacilli, but not much more. Our spirals come in the curved comma-shaped vibrios, a kind of gentle turn with our spirillum, and the spirochetes, which are more of a true cork-shaped tight spiral. Just as a note, the morphology often gives rise to some of the genus names. So seen here, we see bacillus, if capitalized in italics, or it can be underlined, that would indicate the genus name. If we're just talking about the shape, that rod-shaped, then it would be bacillus in lowercase. We'll see similar things for the genus of Staphylococcus. They get their name from that grape-like cluster morphology or arrangement, and the streptococci, which again, from the fact that those cocci are in chains, those genus names have been derived from those morphology and arrangement. Now we're going to look at the structures external to the cell wall. So we're going to talk about things like glycocalyces, flagella, axial filaments, fimbriae, pili, and see their function for the cell. Some of these are going to be virulence factors in helping the cell to cause infection, but we'll take a look at the individual features of these structures. In the picture here, we see the structures labeled in red are found in all bacteria. So the other things are structures in an idealized bacterial cell pictured here. So we'll look at the structures that are external to the cell wall. Note in the picture that the gold layer is the plasma membrane. All cells have to have a plasma membrane, whether they're prokaryote or eukaryote. Then that brownish layer is the cell wall. And so we're going to be talking about structures that are going to be outside of that cell wall. Now we're going to talk about those structures. The glycocalyx is going to be what's referred to as a extracellular polysaccharide, or EPS. This is kind of this jelly-like matrix that is external to the cell wall. Not all cells have a glycocalyx, but when they do, this is often a virulent structure. As this polysaccharide, this is usually fairly sticky. So when we talk about these glycocalyces, they can be in two forms. They can either be a capsule, which is a nice, tight, neatly organized layer, as we see for the picture to the lower right corner. That is Streptococcus pneumoniae, causative agent of what's referred to as pneumococcal pneumonia. They have a very defined capsule, as you see in yellow. Other things have a slime layer, which is less organized and kind of a loose configuration. For those slime layers, think about what happens on your teeth overnight. When you wake up in the morning, you kind of have that slimy feeling to your teeth before you brush them. Well, that's what that slime layer is. It's bacteria that have these extra polysaccharide layers that allow them to attach. From the standpoint of virulence, capsules are really important. They are sticky, so part of infection means organisms have to figure out how to hold on. If we stick with the pneumococcal pneumonia, we see the respiratory system is constantly trying to move things up and out. And so therefore, having this sticky capsule helps them to adhere so that they can possibly cause infection. Capsules also prevent phagocytosis by white blood cells. What this means is that this jelly layer to the outside is kind of like Jell-O. And like Jell-O, if you try to pick it up, it kind of squirts through your fingers. Well, that's the same thing that happens when a white blood cell tries to grab on to a bacteria that has a capsule. But like Jell-O, if you get Jell-O all over your counter, it's really sticky. And so that's why the capsule can be both for adherence and help to prevent phagocytosis. Flagella, on the other hand, are for motility. They are made up of chains of protein that are flagellin, and they make these individual filaments. The filaments need to be attached to the bacterial cell, and that is done through a protein hook. This then gets anchored through the cell wall, so through the peptidoglycan layer, and actually also anchors into the cell membrane. If we look at the pictures to the right-hand side, we see the top figure is of a gram-positive bacteria, and we see that it has what are referred to as basal bodies, two basal bodies for the gram-positive to help it anchor into the plasma membrane as seen in the figure in figure B. The bottom one, which is the A figure, this one is for a gram-negative bacteria. We'll talk about the difference between the cell wall structure for gram-positives and negatives in a moment, but suffice it to say here that gram-negatives have 4 basal bodies that help to anchor them through two membranes and their peptidoglycan cell wall. When we look at the arrangement of flagella, different bacteria have different number of flagella. In panel B, we see the monotrichus, which is polar, meaning it's at one end, and so that single flagella helps the organism move from point A to point B. In panel A, we have what is referred to as the paratrichus. These are kind of a Medusa-like looking organism where they have flagella just all over. Usually these types of bacteria are very motile and often may even swarm even when you're trying to plate them out on agar. In the lower left panel, panel C, the Lophotrichus is polar, but it's a tuft or more than one flagella at one end or pole. Although it's not the same prefix, kind of can remember these by being lopsided since there's only a tuft of flagella at one end. In the last panel, panel D, the Amphitrichus, this has flagella at both ends or poles. So again, not the same prefix, but think about these as being ambidextrous. When people are ambidextrous, they can use both hands. Well, these just have flagella at both ends or pole. When we look at the motility, the flagella actually rotates, and this causes the organism to do a run and tumble type of movement. When I say that a bacteria can get to point A and from point A to point B, it's not usually a straight shot. So they can sense their environment. They can move toward or away a stimulus. Sometimes that's a chemical stimulus, so chemotaxis. Some bacteria can be photosynthetic, so they may have phototaxis. But even when they do so, they do this run and tumble. They make a run where all their flagella are rotating, but then they stop and tumble where they sense their environment. In this way, if you chart their path, yeah, they do get from point A to point B, but they've made several different pathways in their effort to get from one point to another. Because flagella are made of protein, we've also utilized these flagellar proteins to be able to use them for identification. Flagella proteins are referred to as H antigens, and an interesting story for the E. coli, the O157H7, is that this is a unique serovar. That means that it's a variant that is able to be recognized by serological techniques. In this case, we're using antibodies to help us identify the flagella antigen that will indicate the pathogenic strain. E. coli is normally good fecal flora, and so it became problematic when we started to see this pathogenic enterohemorrhagic strain, the O157H7. You may have heard of this in earlier days. The first outbreak was with the Jack in the Box hamburgers out west, and this can be deadly for the fact that it causes this enterohemorrhagic strain, and it can also cause kidney damage. So it was problematic for us in the lab to try to find the pathogenic one in all of the regular E. colis. So using the unique flagella protein for this bacteria, the H7, it helped us to identify the pathogen fairly quickly. There's also some very other interesting research going on with targeting flagella. We all know the problem with antibiotics and their resistance, so when we first think of treating a bacterial infection, we do think antibiotics, but we need to start being more creative. The other problem is that antibiotics kill a lot of bystanders. They can kill good bacteria as well, which we now realize are important for our gut health and our overall health in general. So we don't want to just kill any bacterium, but we would like to target those that are the pathogen. So one strategy that I have provided the link for here, and there's also a video in there so you can see how flagella work, is to target the flagella of these pathogens. If you disrupt their flagella, well, they may not be able to infect as well, find the area where they are trying to actually cause disease, and so therefore this has become a target in trying to fight infection. We also have another type of flagella. It's referred to as a endoflagella or axial filament. Endo because in spirochetes, this flagella is actually twisted in those twists and turns of the corkscrew spirochete. It's anchored at one end, but it allows the spirochete to rotate in this corkscrew manner and get through cells and fluids and mucus, and this often helps it to cause infection. Here we see what that axial filament looks like as it's wrapped around the spirochete. It really is entwined with the spirochete itself. A couple of spirochetes that you may know by disease, but maybe not by the bacterial name. Lyme's disease is caused by Borrelia burgdorferi, which is a spirochete. On the left-hand side, you see the bullseye rash that is often associated with Lyme's disease. And one of our sexually transmitted infections, syphilis, is actually also caused by a spirochete. Treponema pallidum is the causative agent, and you see in the panel there the corkscrew-shaped spirochetes that causes the disease. So looking at other bacterial structures to the outside of the cell wall, the fimbriae allow for attachment. The picture to the right shows Neisseria gonorrhoeae, a diplococci, where it has all these needle-like projections from it. These help the bacteria to adhere. So therefore, it's a factor in pathogenicity. Again, in order to cause infection, one of the first things that needs to happen is the bacteria needs to hold on. For fimbriae, there's usually numerous of these hair-like projections. And again, these are protein filaments, the protein being pillin. In the bottom panel, we see pili. Pili are used to transfer DNA from one cell to another. So there usually are only one or two, and they're much longer. So in the picture, you can see it's forming this channel, this connection between two bacterial cells, and it may allow it to pass genetic material from one to the other. Maybe it is even able to give it genes that are allowing it to be resistant to an antibiotic. For that reason, because of the genetic recombination, these are often referred to as sex pili. Although we do not see sexual reproduction in bacteria, we do see the fact that this is going to be a ability for the organism to get new genes, and that's kind of what sex in eukaryotes is about, generating that genetic diversity. So the next layer is going to be the cell wall. We've referred to already gram-positive and gram-negative bacteria, but now we're actually going to define what that means. So the cell wall itself, the brown layer in the figure, is a semi-rigid structure that gives the characteristic cell shape. Without the cell wall, we wouldn't have cocci and rods and spirals, and so that's very important in their morphology. The cell wall also prevents osmotic lysis. So when we look at the environmental conditions a bacterial cell is going to have to endure, some days it's going to find itself in a nice moist water environment. Other days it may be an environment with more salt or sugar. All this is going to change the osmotic pressure, and the cell wall under certain conditions are going to prevent the osmotic lysis. So the bacteria doesn't want to explode when it's in pure water. The cell wall is uniquely made up of peptidoglycan for bacteria. So that is a defining structure or component of what are considered to be the eubacteria or the true bacteria. So originally, penicillin was called a miracle drug Because it doesn't harm human cells. Why doesn't it? Penicillin was designed to prevent the making of that peptidoglycan cell wall. So it sounded great. Unfortunately, it doesn't work as well on all bacteria. Penicillin works better on gram-positives than gram-negatives. And as soon as we started to use the miracle drug, we also started to see those organisms that were resistant to penicillin. And therefore, penicillin is still a very useful antibiotic, but by no means the miracle drug. However, it does have limited toxicity. And that's the whole point of designing antibiotics, to make sure that you're targeting just the bacteria and not doing harm to the host cell. So by targeting the peptidoglycan, that meant that it wasn't going to harm the eukaryotic host cells. So the peptidoglycan of the bacteria is actually a polymer of two sugars. So it's a polymer of disaccharide. The two are N-acetylglucosamine, NAG, and N-acetylmuramic acid, NAM. Those link together to form the carbohydrate backbone. So it's kind of like the beams of the cell wall. There are also these peptide cross bridges that are just short chains of amino acids that are going to help to link the beams together. And we also see these tetrapeptide side chains that kind of go in the upward direction. These are going to help us to stack on these layers, which is going to be important for gram-positive organisms. Here we see the linkage between the N-acetylglucosamine and the N-acetylmuramic acid. So this just means that this is going to be the repetitive. So this repetitive unit of the NAG and NAM, that's going to make up the main carbohydrate backbone of the cell wall. When we talk about bacterial cell walls, we say that gram-positive cells have a thick peptidoglycan cell wall. If you look at the B figure, the gram-positive cell wall, you have the gold, which is the cell membrane, and then the brown area is that thick peptidoglycan cell wall. If you look over at the right-hand side, you see the cell membrane, the plasma membrane, which is a phospholipid bilayer, and then you see stack upon stack of the peptidoglycan cell wall for a gram-positive. So it has multiple layers of this peptidoglycan, and it has additional support of teichoic acid. These are filamentous types of structures that run through the wall. So we have the wall teichoic acid, and some of them have a lipid component that help them insert into the membrane. So those are the lipoteichoic acids. In the bottom panel, we see that the gram-negative cell wall is more like a sandwich layer. You're going to have two membranes that are going to be like the bread, and then the peptidoglycan is going to be the bologna in between. So if we look over to the right-hand side, we see the plasma membrane, and then there's a periplasmic space that is going to house the peptidoglycan layer. That is only a single monolayer, and it makes up kind of the difference of not having a thick peptidoglycan layer by having an additional outer membrane. This additional outer membrane is referred to as the LPS layer, or the lipopolysaccharide layer. It has these core polysaccharides and the O polysaccharide, which are, again, a feature that can help us in identifying some of the pathogenic organisms. And it also has a lipid A component. The lipid A component is considered a pyrogen. It actually is one of the reasons that gram-negative organisms can cause fever. So we see that the gram-negative is more like a sandwich layer of having the cell membrane and the outer LPS as these phospholipid bilayers, and then the peptidoglycan in between. So if we summarize, The gram-negative cell wall actually only has this thin monolayer of peptidoglycan, but its major feature is the outer membrane that is going to feature those lipopolysaccharides, and so this is going to have the LPS layer. Where the peptidoglycan resides, that's referred to as the periplasmic space. When you think of a gram-positive, you should think of this as having a thick cell wall. That's because it has layer upon layer of that peptidoglycan. It also has teichoic acid running throughout to give it more stability. And we'll talk later about some unique gram-positives. In acid-fast cells, which are a subset of gram-positives, they actually have a waxy component referred to as mycolic acid. We'll now take a look at what this actually means for the cell. So in summary, gram-positive cells, teichoic acid, both wall teichoic acid, lipoteichoic acid. But one of the things at that kind of open weave network of the thick cell wall, it does help to regulate the movement of cations in and out of the cell. It also provides some of the polysaccharide diversity for antigenic variation. One of the ways to think about this is think about a strep test to determine if you have the pathogenic Streptococcus pyogenes. We have developed antibodies to allow us to recognize this antigenic variation, and part of that is coming from this polysaccharide difference that different gram-positives will have that aids us in being able to test for the pathogens. When we talk about the gram-negative, things to remember: that LPS layer, so that lipopolysaccharide, but in general they just have more lipoproteins, and because they have an extra phospholipid bilayer, more phospholipids. They have the unique periplasm between the two membranes, and interestingly, that extra membrane layer with the LPS helps prevent it from phagocytosis. It again is kind of this protective barrier of both sugar and lipids that prevents white cells from being able to grab on. It also is just another barrier that prevents things like antibiotics and complement from getting in. Complement is an immune component that is going to allow the lysis of bacterial cells. So preventing that complement from getting in, that extra outer membrane is just a protective barrier. We also see the O-polysaccharide antigen revisited in our E. coli O157H7. So again, the pathogenic form of the E. coli, the enterohemorrhagic strain, we use not only the flagella antigen, the H7, but the unique O157 O-polysaccharide in helping us to recognize that pathogen. And as I said, lipid A is something that causes fever. It's a pyrogen, but it's referred to as an endotoxin. So it has toxic properties that are going to cause potentially things like endotoxic shock, which we'll talk about later in the term. Because it has this extra layer, it also needs to have these porins. These pores are basically channels through the membrane, and these are going to be proteins that are embedded to help things to be able to get through that extra layer. So the gram-negative, again, we see the picture here. It's kind of more the sandwich layer, cell membrane, peptidoglycan, the bologna in the middle, and that outer membrane which contains the LPS layer. So we've defined these as gram-positive and gram-negative, but what does that really mean? Gram staining is a differential stain that allows us to get the gram-positives to be one color, blue, purple, and the gram-negatives to be red after we do the staining procedure. The gram staining procedure utilizes basic dyes. Basic dyes just have a positive charge, so they're going to be attracted to the cell. Cells in general usually have a negative charge inside due to the DNA and other negatively charged proteins. So dyes that have a positive charge are usually going to be attracted and stain the cell. So the differential part is that we're going to use two different stains to be able to see the two different types, gram-positive and gram-negative. When you're doing a bacterial stain, the first thing you have to do is make a smear of bacteria and heat fix it to the slide. Here we see, once you've done that, you then apply the first primary dye, which is crystal violet. As the name implies, this is a purple dye. This is going to turn everything purple. The next one is the application of a mordant or a fixative, what's referred to as grams iodine. As the name implies, this is going to help fix the dye into the cells. But the difference is going to be in the cell wall structure between gram-positives and gram-negatives that are going to give us a differential feature. The differential step is going to be the decolorization step with the alcohol wash. The alcohol is going to decolorize the gram-negatives, so they would be colorless at this point, but the gram-positives remain purple. When we want to then visualize, we can't see that colorless cell, so we apply a second dye or the counter stain, in this case using safranin, which is a bright red color. So in the end, gram-positive bacteria are going to be a bluish purple color, and gram-negative bacteria are going to be red. The principle behind this is that when the crystal violet and iodine are mixed together inside the cell, they form a crystallized structure. It's now bigger and more difficult for that to get out of the cell, but the different cell properties between gram-positive and gram-negatives determine whether that crystal violet can be washed out or not. Gram-positive, the positive does not refer to charge. It refers to retention of the primary dye. So when we talk about a gram-positive, they're going to retain the primary dye throughout. When we talk about a gram-negative, they're going to lose the primary dye and become colorless, and that's why they have to be counter-stained with the safranin. So when we fix our bacteria to the slide, at first both of them do not have any color, and then we add the crystal violet. The crystal violet is going to go into both cell types, so we see both gram-positives and gram-negatives would be colored purple. Then as we add the grams iodine, they turn more of a violet color. Here we see that the gram-positives are going to be retaining and the gram-negatives are going to be losing the dye in the next differential step, which is the decolorization. When you add alcohol to the gram-positive, alcohol dehydrates cells. So the peptidoglycan starts to collapse and become more dense. What that means is that it's not as porous, so the crystal violet iodine crystals get trapped in the cell and don't leave. In contrast, the gram-negatives, alcohol dissolves lipids. So alcohol begins to dissolve that outer LPS membrane and then leaves just that thin monolayer of peptidoglycan. It's very thin and fragile, so without the protective membrane, that cell wall begins to fragment, and the crystal violet iodine complex then easily washes out, leaving the cell decolorized. And if we looked underneath the microscope, we wouldn't be able to see it. So then the last step is going to be the counter stain. The counter stain is not going to displace the crystal violet that is already in the gram-positive, so the primary dye gets retained throughout, but then the crystal violet is not retained for the gram-negative. They lose that primary dye. So in the counter stain with the safranin, they take up the red dye, and hence our gram-positives are going to be blue or purple, and our gram-negatives are going to be a nice red color. So our gram-positive cell wall, 2-ring basal body for the flagella. Things that we'll look at is that the types of things like penicillin and lysozyme that disrupt peptidoglycan are going to be more effective against gram-positives. The gram-negative cell wall, 4-ring basal body for its flagella that anchors it into the cell wall. It also, again, is toxic, so it is the endotoxin because it's part of the cell. And we use different antibiotics for gram-positives versus gram-negatives. So tetracycline is usually the treatment of choice for gram-negative cells. Below, we also see some gram-positive bacteria and gram-negative bacteria that you may recognize. So gram-positive bacteria, Staphylococcus aureus, Clostridium botulinum, causative agent of anthrax, Bacillus anthracis, Streptococcus pyogenes, causative agent of strep throat, just to name a few. The gram-negatives are E. coli. Escherichia coli is always our go-to gram-negative, but we see some other ones that maybe you're familiar with. Neisseria meningitidis is the cause of what is referred to as meningococcal meningitis, and things like Neisseria gonorrhoeae, the causative agent of gonorrhea, and Helicobacter pylori, causative agent of ulcers. So these are just examples of some of the gram-positive and gram-negatives that we'll start discussing throughout the term. So if we look at a summary of gram-positives versus gram-negatives, we see gram reaction. Gram-positives retain the crystal violet and iodine complex, so they are going to be purple at the end of the staining procedure. Gram-negatives are decolorized and accept the counter stain and are stained red. The peptidoglycan for gram-positives, nice and thick, multi-layered. For the gram-negatives, it's a thin monolayer in between those two membranes. Only gram-positives have teichoic acids to help stabilize the thick peptidoglycan, but gram-negatives have the periplasmic space. They also, because they have two membranes, they have the outer membrane, and they also have in that outer membrane, that is the LPS layer. So high lipopolysaccharide content, and in general, gram-negatives have a higher lipid and lipoprotein content due to that outer membrane. Other features, gram-positives, as we said, the flagella structure, 2 rings in their basal body. For gram-negatives, 4 rings. The toxins produced are different. For gram-negatives, the toxin is predominantly endotoxin, part of that LPS layer, as we will see, endo meaning in, or in this case, part of. For gram-positive, those, they primarily produce exotoxins. Exotoxins are secreted from cells, exo meaning out, and we see some of the most potent toxins from things like Clostridium tetani, causative agent of tetanus. So it's this potent exotoxin that causes the rigidity that's associated with the disease tetanus. We also see that gram-positives are more likely to be on the skin and in the environment because they're more resistant to physical disruption. That is because they have that nice thick peptidoglycan layer, and that also means that things that disrupt that peptidoglycan, like lysozyme, that means that they're going to be more susceptible to things like lysozyme. For antibiotics, gram-positives are more susceptible to penicillin and sulfa drugs. For gram-negatives, this is going to usually be antibiotics like chloramphenicol and tricycline. There's also an old-time treatment of gram-positive bacteria, inhibition by basic dyes. If you had a cut or scratch, you could actually take a dye like crystal violet and paint it over the area to prevent infection from gram-positives. We also see gram-positives are susceptible to anionic detergents. So when we wash our dishes and things, we're mostly getting our normal gram-positive flora on there, so that makes sense. Resistance to sodium azide. This sodium azide is an inhibitor of the electron transport chain. It is easy for it to get into the gram-positive, but not into gram-negatives, so we often include it into some of our solutions. But when we talk about gram-positives, their resistance to sodium azide is high, but it is fairly low for gram-negatives. Gram-positives are also more resistant to drying. They are better equipped to be out in the environment because of their peptidoglycan thick layer. For gram-negatives, the nature of that membrane means they need moisture. So same way as if you leave jelly out, that jelly dries. Well, that's the same thing that happens to a gram-negative. So gram-negatives are less resistant to drying, so they don't survive as long out in the general environment. We're now going to look at some atypical cell walls. So there are some organisms that are referred to as mycoplasms that are the smallest bacteria that can grow and reproduce outside of a host cell, but they're unique in that they lack a cell wall at all. They have sterols in their plasma membrane, things like cholesterol, to kind of make the membrane more rigid so they can survive. Think of walking pneumonia. It is actually caused by mycoplasma. So that is an example of one of these organisms that you may not have been aware of. As I said before, the archaea bacteria are prokaryotic, but they're usually wall-less or have walls of sodium urine. They don't have peptidoglycan. The archaea bacteria were considered to be the most ancient, at least we thought so. That's because they were found in extreme environments like very high temperature environments and methane environments that might have been indicative of some early environments in evolution. However, when we look at the tree, we actually see the archaea and the true bacteria split off fairly early in evolution. So the archaea bacteria are more related to the eukaryotes than they are to the other typical bacteria. We also see a clinical relevant atypical cell wall in acid-fast cells. You have heard of tuberculosis and leprosy. Well, those are caused by Mycobacterium tuberculosis and Mycobacterium leprae. These types of cells are subsets of gram-positives, but they have a high concentration of mycolic acid. This is a very waxy substance, so they don't stain very well in the gram stain procedure. In order to stain them, you have to do an acid-fast stain. So you take this bright pink dye, carbofuchsin, but you have to heat it to be able to melt the waxy substance to get the carbofuchsin into the cell. After you do that, you then decolorize with what's referred to as acid alcohol. This acid alcohol is acidic, but it's also kind of cold, and so it causes that wax to solidify again, trapping or holding fast the dye inside. So the acid-fast cells remain a bright pink color, and non-acid-fast cells are then decolorized. We then usually use methylene blue to be able to look at the non-acid-fast cells. So when you look at a sample, you see these bright pink acid-fast cells and these blue non-acid-fast organisms. We can also do things to damage cell walls. Some of this is natural, some of this are things that we introduce. So lysozyme is present in all your secretions. One of the things that it does is digest the disaccharide in the peptidoglycan layer. So it's helping to control that a lot of your normal gram-positive flora doesn't get into the wrong place. Penicillin, as we talked about before, was this wonder drug because we thought, aha, it's going to affect only the peptidoglycan layer unique to bacteria because it inhibits the peptide bridges, but it's not universally effective. Most gram-negative bacteria are not as susceptible because of their outer membrane. We also like to play around with cell walls, so we can make a wall-less cell, and if we do something like treat with lysozyme, we can turn that cell into a protoplast. If you don't have the cell wall, you don't have the characteristic feature of what the morphology looks like, and so the cell doesn't replicate normally. A spheroplast is a wall-less gram-negative cell, but both protoplasts and spheroplasts are very susceptible to osmotic lysis without that cell wall present. Over time, they often turn into what are referred to as these L forms, these wall-less cells that start to get abnormal. They start to pinch off and bleed part of their cytoplasm and membranes. So these are ways that we can study the features of the cell wall and also try to study the cell membrane, as we'll see in the next slideshow. So we've covered our prokaryotic and eukaryotic cells, and we just want to make sure that you check through